Hello, everyone, and welcome to today's presentation titled Modeling Inflammation and Fibrosis in Humans with PSC-derived Steatohepatitis liver organoids, presented by Takanori Takebe, Associate Director of the Center for Stem Cell and Organoid Medicine and Professor at Cincinnati Children's Hospital, as well as Professor at Tokyo Medical and Dental University and Professor and Principal Investigator at Yokohama City University. My name is Mark Kennedy of Thermo Fisher Scientific, and I'll be your moderator for today's presentation. So before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting any questions you may have at any time during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your question into the drop-down box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. So if you have any trouble seeing or hearing the presentation, please use the Ask a Question box to let us know of any technical difficulties. So with that, please allow me to introduce Dr. Takebe. So Dr. Takebe is an Associate Director of the Center for Stem Cell and Organoid Medicine, also known as CUSTOM, at the Cincinnati Children's Hospital Medical Center. And as I mentioned, he's also Professor at Yokohama City University and Tokyo Medical and Dental University in Japan. He serves on the Board of Directors at the International Society of Stem Cell Research, and his lab investigates the mechanisms of human organogenesis, and he develops mini organ technologies from human stem cells, namely organ bud-based approaches. He is applying iPSC liver buds into drug discovery as well as transplant applications for patients with a rare congenital metabolic disorder, ultimately expanding the clinical indications to diseases like liver cirrhosis. And with that, I'd like to thank Dr. Takebe for joining us, and I will now turn the presentation over to him.
Thank you so much for the kind introduction, and good evening, I think, to everyone. And I'm in Japan, and good morning from Japan. And my name is Takebe, and thank you so much for Thermo Fisher members for giving me this really high-tech opportunity to present our recent work related to organoid. So I just slightly modified my presentation title, so the era of organoid medicine, to cover the more broader perspective of what we are working on using the human pluripotent stem cells. All right, so I just a little bit about great thanks to my members in the different labs. So I'm currently running 5 different laboratories in Cincinnati Children's, in Yokohama City University in the middle, and Takeda joint program in the Takeda Pharmaceutical and Design Center, which is a little different from my talk, and also Tokyo Medical Dental University. So especially I like to appreciate Dr. Taniguchi, who is also the professor at Yokohama City University, and we are working very closely for the translational application of our liver organoid-based approaches. And also I'd like to thank for the funding agencies, including the AMED, GST, GSPS, and NICEF, and Takeda Pharmaceutical as well. And also collaborator as well. So I'm working in a Cincinnati Children's at CUSTOM, and I'm really grateful for the tremendous support from the CUSTOM faculty members at Cincinnati Children's. And also Shinya Yamanaka, working at the Kyoto University, is providing us the fantastic stem cell source, which is namely on iPS cells for the translational applications. And with that, I'd like to start my presentation. So just as a way of background, so I'm originally interested in transplant surgery, especially for the liver transplantation. So this is the first patient I actually met in the United States when I was taking the internship at the Columbia University. So as I said, here is U.S., but the patient I met there was a Japanese patient who is Mr. Asano. And he was very lucky guy because he was diagnosed as a very severe liver failure back in Japan. But unfortunately, there are a very limited number of transplants in Japan probably because of limited gunshots. So his only option was to fly outside Japan get a whole liver transplantation in elsewhere outside Japan, and he was just flying into the Columbia University, and finally be able to be transplanted by the whole liver transplant approaches. And now he's completely recovering from the disease and back in Japan and working as an elementary school teacher as before. So he was very lucky to be transplanted. But I think behind the scenes, I think we are missing the very severe statistics like this one. So this is the statistics from the United States and for the transplantations. So as you see from the blue bar, starting from the early 1990s, the demand or waiting patients for the whole organ transplantation is growing over time very intensively. But in contrast, actual transplant case indicated by green bar, it remains stable, which simply suggested that the gap is growing and almost hundreds of thousands of patients are just waiting to die. So this is really catastrophic and very sad. So I decided to jump into the different field, which is namely a stem cell and organoid field, to make a difference for the patients waiting for the transplantation. So to do that, we are very interested in the organoid technologies. So this slide just show you the review papers published by Nature, authored by the Cassandra, and back in 2015. And as you see here, there are a number of different organoid technologies are coming out for the past few years, including the brain organoid and liver organoid and gastric organoid and so forth. And the Dr. Sasai, working at the Riken, is I think the one of the founder for this field by publishing a paper for the optic cup organoid paper. And also Hans Krieber's lab and Dr. Toshi Sato is developing a method for developing a gastric organoid from the adult stem cell populations, followed by many outstanding investigators in the world to develop multiple different organs. I just want to review the background of the evolution of the organoid field in the next slide. So organoid is initially defined in early 90s. And growing steadily over time, but for the past few years, the number of fabrications for organoid is growing intensively, as in the slide. So I think throughout the evolution process, I think very roughly summarizing, I think there are three major contributing technologies driving the organoid evolution field. So for instance, if you look at the organoid 1.0 version 1, I think this is the initial foundation of the organoid technologies. So Dr. Wilson is working with the sea sponge, sea animals, and found that if you dissociated the sea sponge into the single cells and reaggregating them in a small well, he found the entire sea sponge structures can be reformed by the reaggregation technologies. So I think reaggregation found in the early 90s is clearly a very important technologies to look at the self-organization potential, which is a critical aspects for making an organoid. But afterwards, it turns out the more higher animals like rodent models or monkey model, I think still it's very challenging to make an organoid from just an aggregate. And to tackle this, Mina Bissell and other pioneering scientists using the laminin-rich extracellular matrix component, or in general known as matrigels, is really important for enabling the self-organization into organoid. So combining with the reaggregate with the matrigels or laminin-rich ECM, she and many of the investigators found out even for the rodent model, we can make an organoid, including the mammary organoid or kidney organoid or lung organoid. And as a version 3, I think this is the major driver for the organoid increase in organoid publication is the human stem cell technologies. So including the adult stem cells and pluripotent stem cells, including the embryonic stem cells or iPS cells. So having that adult stem cell source or pluripotent stem cell source, Dr. Sasai and Dr. Trevers found out the method to make a human-specific organoid. And this was followed by a series of investigators, including Jim Wells and Dr. Taniguchi and Dr. Novri and Nishi Nakamura, Marisa Little, to make her gut organoid or kidney organoid or brain organoid and so forth. But today I'm going to really focus on the future of organoid, which I think the organoid medicine or application of organoid for the medical application. So for the past 3 version of organoid evolution was really driven by the method to make an organoid. But it turns out we are now able to make a sort of human miniature version of organ. So I think we, for the future, I think I'm really interested in the promise of application of organoid into the medical field. So to accelerate this process, at Cincinnati Children's, we are recently established our new center called CUSTOM, Center for Stem Cell and Organoid Medicine. So Alan Zorn, who is a noted developmental biologist in a liver field or lung field, is a director for the CUSTOM. And co-directed by James Wells and Mike Helmuth and myself. And Jim Wells is working on a number of GI organoid, including gastric or intestinal or large intestinal organoid as well. And Mike Helmuth is a surgeon and clinician scientist and developing a method to transplant the intestinal organoid to model the human development in vivo. So we are establishing very critical infrastructures to increase the access to the organoid technologies, including the core laboratories, including a gene editing core, or imaging core, or epigenetics core, and so forth, and to help the clinician scientists to use the very high-tech approaches for the organoid research. And also we are developing a blanket ILB, which enables the collection of the human iPS cells or eventually to develop organoid as a banking strategies from the rare disease patients as well as healthy kid patient populations. And also we are developing accelerator laboratories, which is, I think, very unique infrastructures to enable the interaction between the academia and industry. So we are welcoming the industry scientists with a close proximity to our academic laboratories to be able to, for instance, screening the drugs in our organoid system for the commercial prototyping. And also we have the clinical grade GMP laboratory for the tissue processing in a clinical level. So these sort of 4 major accelerator components is helping the scientists to use the organoid for the medical applications. And we are actually dividing the 4 different phases for the eventual development of organoid medicine. So always, I think the important thing is the basic science part. So we are studying the human development and modeling the development in vitro, or we can make in complex organoid structures. So we have still many challenges, which I am going to highlight in a later slide. So we still clearly need basic science components as a major driver. But I think equally important, the prototyping as a second phase is also important. So for instance, I think if you use the organoid for the drug screening, I think we need to show the positive evidence to predict the human clinical trials, and also negative evidence to predict the human outcomes, which, for instance, the drugs failed in the clinical studies has to be recapitulated in disease organ conditions. And so I think we need to show the sort of preliminary evidence for the organoid-based application as a prototyping stage. And if you see the very promising potential, I think we can move forward or move further for the translation. So we are currently working on the transplant application of liver organoid. So we are working with the large-scale production protocols or larger animal model to test the efficacy or safety in preclinical conditions to move forward. And also some of the commercial prototyping has a potential for the industrializations. So some case, I think we are developing a startups, and some case we are licensing out to the companies to facilitate the commercial application of organoid for improving the accessibility for the many scientists or clinicians to use organoid-based approaches. So I'm jumping into the basic science part, which I'm learning in my laboratory in Cincinnati. So the questions we are working at Cincinnati Children's is very simple. So how we can know the process of organogenesis and how we model that complex process by using the human stem cell culture system. So this is the top questions we are still dealing with. So, the overall, I think the organoid is really driven by the basic developmental biology. So I would say translational embryology is really key for the organoid development. So, the gastrulation is the initiation of the human development or every animal development. And afterwards, the tissues are specified into the progenital stage, including endoderm or ectoderm or mesoderm. So we are particularly interested in the foregut endoderm, which is diverse from the early primitive endoderm structures. So foregut endoderm is really giving rise to a variety of different organs, including the thyroid, esophagus, and lung, and so forth. But we are specifically interested in the border between the foregut and midgut and posterior foregut portion is giving rise to the number of critical organs for the metabolism, including liver tissues, biliary tissues, or ventral pancreatic population, or dorsal pancreas as well. So what we are working on is using the human stem cells to model the molecular logics, which is identify the classical developmental biology to leverage that in a stem cell culture model to develop a design principles for recapitulating the human organogenesis in the dish. So back in 2013, we are initially focusing on the most primitive process of liver bud formation or early liver development. So early liver bud is actually specified from the part of posterior foregut tube. So at this stage, the population is named hepatic specified endodermal cells, and this is about E95, just before E95 in the mouse, or 1 month after gestation in humans. And hepatic specified endoderm cells at this stage is two-dimensional, sheet-like structures. But afterwards, they are forming a liver bud structures, which is clearly a 3D and very complex structures. And interestingly, these 2D to 3D transitions is requiring the support by the additional supporting lineages, including the endothelial cells and also the mesenchymal populations, which is really found by the Dr. Ken Zawad groups. And once this liver bud is formed in 3D conditions, the blood perfusion is initiated afterwards. And this is really critical for pushing the cells to be more mature and finally developing a complete mature functional liver tissue structures. So what we are hypothesizing is quite simple. So can we model the 2D to 3D transitions by recapitulating these main three different progenitor interactions in vitro in culture? And if that is true, can we push the maturation of in vitro derived organ bud in vivo by perfusing the structure from the circulations in vivo? So this is basically a two-step hypothesis. So human iPS cells directly differentiated into the multiple progenitors and mixed in well to see if we can make her self-organized organ, but in vitro. And if we can make it, can we transplant into the animal model to see if we can get a mature tissues in vivo? So next slide is a simple summary of what we are working on initially. So working with the human iPS cells under FIDA flea provided by Dr. Shinya Yamanaka, which is actually clinical grade. So or directly differentiated into the three major lineages in vitro. I think that, to the right, I think I'm just showing the animation or movies for the self-organization process, but it's not really coming up. But anyway, using the three different lineages, including the transitional hepatic endodome population, primarily marked by the TBX3, and septum mesenchyme population marked by the WT1, and primitive endothelial cell population marked by CD31 or BE calhoun, we are able to make three critical ingredients for the liver bud formations. And using that three progenitor population, we are able to make 3D miniature liver bud structures in vitro. So the next slide is showing the structure of these in vitro liver bud-like structures. So in this pictures, left is human endothelial cells differentiated from the iPS cells, and the green one is a hepatic progenitor population made by the human iPS cells. And this is not really just a simple cluster. If this is aggregate, you want to see the very sparse structures, but it's not. So they are developing network-like structures from the red-marked endothelial cell populations, and aligned with these beautiful network-like structures, we see the hepatic cells are aligned in a beautiful manner. So this is clearly a sign of self-organizations. So we term these structures as vascularized liver bud organoid made by a vitro 3D culture system. So we also characterize by tissue sections or single cell analysis and so forth to confirm these structures is really close to the in vivo liver bud-like tissues of in vivo. But there are critical signs of self-organization like blood vessel networks. We are expecting these tissues can be transplanted in vivo. So the next slide shows the in vivo outcomes. There you go. Okay, so this is the transplanted liver bud tissues. And this whitish part is about 1 centimeter in size, and this is day 0. After 2 days or 3 days, entire part get red, which really suggesting the blood cells are perfusing the entire structures made by the human iPS cells in the mouse of immunodeficient background. So once the blood perfusion is initiated after 2 or 3 days or 48 hours or 72 hours, the blood vessels are remodeled over time to form a more sophisticated blood vessel structures. So the next slide is confirming the actual blood vessel network. So in this slide, the dextran made by green one or blue one is the blood perfusion in the mouse, and red and green, red and orange is the human cell component. So if you're zooming up the tissues, I think it's not coming out. Oh, okay. So this is the to the right is the human iPS endothelial networks. So this is just to confirm entire vascular structures are formed by the human cells or human iPS cells, but blood perfusion was initiated by the mouse circulations. So I originally thought the to the right, I think that has to be animations for confirming the chimeric vessel formation between the human vessels and mouse vessels. But it's not coming out, so I'm just skipping into the next slide. Okay. I think there are animation as well. So this is just to show you the transplant process of liver organoid into the animal model, but it's not coming up. So I'm skipping this. And to the bottom, this is a transplant liver butt. Okay, so this is the number of liver butt are transplanted onto the mesenteric locations, which is close to the portal vein. And to the right, this is the Kaplan-Meier survival curve, and the sham group is as shown here, indicated by blue bar. And after 30 days, most of the mouse is dying over time after 30 days. But if you transplant the human liver organoid into this location, or mesenteric transplantations is rescuing the liver failure, or which is lethal, indicated by red color. So this really simply suggesting that the most of the surviving mouse are rescued by the functional replacement by the liver transplantations, and this is a therapeutic prototype of the concept of the liver organoid transplantations. But I think this approach is really, I think, universal or generalizable for the other tissue as well. So we are working on the other tissue organoid model as well, including the gut tissues, lung tissues, kidney tissues, or cardiac or brain, and also disease tissues like cancer. So our 3D culture approach is enabling entire tissues to be vascularized in vitro and to form a 1 centimeter tissues in vitro. And if you transplant into the mouse using the human cells, again, we are able to vascularize after 48 hours of transplantation. And these vessels are remodeled over time as well. And I think one of the most remarkable organs was the kidney, because I think kidney has a very tremendous or powerful potential for the in vivo self-organizations. So this is just a macroscopic confocal live imaging analysis of generated kidney butt transplant. And green marks the mouse kidney cells, and red marks the blood vessels. So combined, as you see here in the middle, I think very small, tiny glomeruli-like structures are beautifully formed from the scratch. And this is a really, I think, remarkable aspect of the organ butt transplantation approaches. And also, we are working on a pancreatic model as well. And as you see here, to the left is also the movie time-lapse movie for the pancreatic vascularized organ inflammation process. But again, I don't really see any videos, so I can skip here down as well. And this is the middle is the macroscopic view of the generated vascularized pancreatic islets, and that can be transplanted into the capsule of the kidney. And using that transplantation model, we are comparing the therapeutic potential against the fulminant diabetes model in the mouse. So to the bottom, Kaplan-Meier again, survival curve showing the fulminant diabetes is really severe, because after 4 or 5 days, without transplantation, most of the mouse is just dying. But if you transplant the mouse primary islet, indicated by blue bar, this transplant approach is effective to some extent. So almost 50% of the mouse is recovering from the fulminant diabetes if you transplant the mouse islets. But I think the islet transplantation is very promising, and because of the promise, it's actually tested in human or clinics as well. But the problem is the vascularization or early engraftment. So islet transplantation is lacking the vasculatures. So if you combine with our organ-based approaches, we are expecting that transplant efficiency might be improved. And because of that, indicated by red bar, the vascularized islet transplantation using our organ butt culture approaches is really improving the efficacy of islet transplantations, as indicated by the red bar. And this simply suggesting the transplant efficiency is really important for the therapeutic potential against the diabetes conditions. So having that proof of concept demonstrations, we are now primarily working on the liver organoid transplantation to rescue the diseased patients. And final part of my basic science is, I think, the next very big challenge in the organoid field. So currently, organoid scientist is just looking at the single organ to generate. So in our case, we are working on the liver tissue generation from the scratch or stem cells. But I would say I think the single organ generation approach is quite limited in terms of the longer in vitro maturation or longer in vivo functionalization. Because if you have a defect in the surrounding structures like biliary portion or bile duct or pancreatic ducts, I think we have liver failed or we have damaged the liver tissues in vivo in a critical conditions. So, the sort of inter-organ connections or multi-organ connections might be, I think, the next basic science challenge to go forward. So Hiro Koike, working in my laboratory in Cincinnati, spearheaded this program. So he's working on the more complex organoid model, and he recently found so this is a reporter human iPS line labeling the human PROX1 genes by RFP. So using this reporter system, he was able to, from the posterior foregut, to make connective structures, including the liver tissues, as well as a biliary portion or duodenum portion as well. So to the right, this is the whole multi-immuno staining of the PROX1, which is a hepatic portion, and DVA is the biliary portion. So combined with two colors, we are able to show the functional connection between the liver and biliary tracts. And also we have a pancreatic to biliary connections as well as the biliary to intestinal connection as well. So this is a microscopic view of in vitro derived human organoid as well as a mouse real in vivo structures, which is closely resembling the human iPS organoid-based approaches. So I think this kind of proof of concept approach is really important for looking at the expanding the potential for other organs as well, like kidney, ureter, bladder connections, or central nervous system to the peripheral nervous system connections, or lung to airway connections, and so forth. So I think this is a sort of very important challenge to go forward. So this is just a basic science part of laboratory, but from now I'm switching a little bit for the translational applications, namely on organoid medicine. So, the organoid can be developed from a patient-specific stem cells or a healthy person-derived stem cells. So using that tissues, I think we can study the human-specific biology as a basic science, but more importantly, I think we can use these tissues as avatar to the human disease conditions. So having that avatar, I think we are able to monitor the disease progressions or disease prevention or disease treatment in vitro. So having that approaches, I think we can envision the precision medicine application or drug testing application in pharmaceutical industries, and ultimately using a healthier stem cell organoid, I think we can transplant back to the patient to see if we can rescue the disease conditions. So we can foresee a number of different applications using our organoid-based approaches. And my lab particularly interested in the liver organoid, so we initially targeting the application to the drug development process. The hepatotoxicity is a very bottleneck or critical hurdle for the drug development process, especially in the later stage of clinical trials. If you have some side effects related to the liver toxicity, I think this will be causing a tremendous loss of the money and also the time for the development. So if you can predict the hepatotoxicity in a human-specific condition, I think that would be really tremendous for the drug development process. And as a phase 2, as a next step, I think we can also use not just a safety screening, but also we can use the tissue for the efficacy screening, such as steatohepatitis, which is a growing medical problems for the number of different countries. And also we can use the tissues for the potentially for the nutritional screening for the preventions. And also we can use the extracorporeal or device to be integrated by the organoid to use the tissues as a sort of supporting device for the liver failure. But as a phase 3 or last phase, I think we can use tissue for the transplantations. We are currently targeting the congenital metabolic error of the patients to be treated by organ transplantations. So I would focus on the two main applications in my talk. The first one is the drug efficacy screening. So as I said, I think the steatohepatitis or non-alcoholic steatohepatitis is really a growing concern in many different countries. So particularly in the United States, I think Nash or steatohepatitis is now a leading cause for the liver transplantations. So clearly, we need a high volume medical needs to be treated by some medications. But currently, there are surprisingly, no available effective drugs to treat the Nash conditions, although they are estimated to be opening up a huge market for the future drug industries. I think this is partly because we are lacking an effective human model to recapitulate the steatohepatitis condition in vitro. So we are hoping to use organoid to model the Nash conditions in vitro. So we spearheaded this problem as well. So initially, we has developed a method to make a multicellular organoid model, including the innate immunity population or macrophage population can be co-developed in liver organoid conditions. So having the immune components, so she hypothesized to see if we can recapitulate the entire process of the steatohepatitis. So steatohepatitis is initially initiated upon the hepatocytes lipid accumulation, or namely steatosis. But if you get the lipid accumulated over time, I think they are activating somehow the inflammatory process triggered by the macrophage or liver-resident macrophage like Kupffer cell. And after the inflammatory stimuli continues, I think the hepatocyte is getting stressed out, and the cytoskeleton is disorganized, and the mitochondrials are overloaded, and eventually developed by the fibrotic population is expanded, leading to the cirrhosis. So we expose a number of ingredients to trigger the steatosis as well as the inflammation, and to see if we can make a fibrotic condition in vitro by continued lipid stimulation in vitro. So this is just a basic organoid model we are currently working on. So we have I think this should be also moving for the liver organ formation process. So we are revising the organoid model from the human iPS cells to model the microanatomy of the human liver tissues. So we have biliary canaliculi structures, which is very important for the bile acid secretions, and we have biotransporters polarized into the lumen of the human liver organoid, and we have tight junction proteins as well. And this is really close to the actual in vivo biocanalicular structures indicated by EM analysis. And having that organoid tissues, I think we are able to look at the bile acid transport visualized by the fluorescent bile acid method based assays. So we are able to visualize by live imaging to monitor the bile acid excretion pathways in an organoid level. So this is very, I think, important for the multiple liver-based applications. And this organoid model has a very unique advantage because we have multicellular configurations. So the majority of cells are composed by the human hepatocyte indicated by the EP count. And this is epithelial cell markers. So 80% actually the hepatocytes. But out of the 20%, I think the half of the population is AL CAM outcome positive or CD166 positive steroid-like cell lineages, which is a mesenchymal cell population residing in liver tissues. And to the bottom, I think less minor population, but we do see the reproducible or EMR1 positive Kupffer-like cell population is also co-developed in these liver organoid structures. So to the right, this is a whole mount immunostaining of the single liver organoid, and as you see here, majority is EpCAM or E-cadherin or CEBP alpha positive hepatocyte populations. But some of the cells are dimension positive mesenchymal cell population responsible for eventual fibrosis, as well as the CD68 macrophage population is co-developed in these liver organoid structures. So having that multicellular configuration next expose fat or carbohydrates in vitro to see if we can induce the steatosis. So to the left, I think this is also the live imaging pathology-like analysis in an organ level. So if you expose the fat or carbohydrates in a higher level, we are able to show the massive accumulation of triglyceride indicated by the green one in the bottom figure. And this is really dose dependent. So if you do not really modify the culture media, I think they never get any triglyceride accumulated in. But if you increase the dose of fatty acid as well as the carbohydrates, we are able to accumulate extensively into the liver organoid. If you continue this lipid accumulated conditions, we are able to show the preliminary fibrosis-like phenotype in vitro. So panel H is the muscle trichrome staining, and after 7 days, we see some collagen depositions. And then the panel I is also the immunostaining of the whole mount tissue analysis. And green one is a fibrotic population, and red one is a epithelial cell population. So combined, I think we see the fibrotic cell population are propagated in our organoid model. And panel J is a biochemical analysis of the supernatant. So this is a procollagen peptide, which is also used in the clinics to diagnose the fibrosis. And this biomarker is also increased in our steatohepatitis-like organoid culture media. So combined, I think organoid is probably able to model the preliminary aspects of the steatohepatitis or fibrosis in the human-specific conditions. But how much predictive in this organoid model? So to test that, I think we are using the genetic contribution to be predicted in an organoid model. So to test that, we are looking at the 3 major variants reported by others, published in the Nature Genetics back in 2017, last year. So using these 3 major, most significant variants, we are stratifying or made a ranking of 2,500 of genotype available stem cell lines, and made a risk stratified panel. So high risk patient stem cell lines and middle risk patient stem cell lines, or minimal risk or low risk stem cell lines, to see if we can model that genetic conditions in a selected couple of stem cell lines. And to the right, this is a triglyceride staining in an organoid level using the different risk patient populations. And as you see here, high-risk patient stem cell organoid is accumulating the triglyceride much higher extent than the middle-risk stem cell or low-risk stem cell-derived organoid. So this really is simply suggesting that genetic factors identified by the 100,000 of patient genotype analysis can be predicted by the couple of different stem cell line analysis in an organ level. So this is, I think, very remarkable to predict the phenotype in humans using the couple of stem cell lines. So having that correlation in the humans, we now test the clinical trials light condition in vitro using the currently testing compounds like obeticholic acid as well as FGF19. So to the left, this is like efficacy analysis, like Kaplan-Meier survival curve. But this is an organoid survival curve. So if you look at the blue or light blue bar, this is a non-drug treated condition. So this is like a sham group. So organoid is eventually dying off over time. But if you expose the obeticholic acid or FGF19, which is remarkably improving the survival after the steatosis stimuli, as indicated by the pink as well as dark gray. And if you do not really expose the fat, I think they are surviving over time. So this really highlighting the organoid-based survival outcomes is somehow similar to the clinical trials tested in phase 3 for the Nash. But not just for the efficacy-like analysis, I think we can use the surviving organoid for the further mechanistic studies. So for instance, this is comparing the reactive oxygen species, which is also the trigger for the inflammatory process in the hepatocytes. So we see the massive accumulation of reactive oxygen species in a fat-treated organoid, but that can be improved or decreased by the obeticholic acid treatment or FGF19 treatment as well. And this is a quantitative analysis. And also, I'm not really going into detail because of the time limitations, but fibrosis can be predicted by the stiffness measurements per organoid. And if you look at the steatohepatitis organoid, the untreated one is really low, but if you increase the fibrosis-like conditions in the organoid, the stiffness is really highly upregulated, as indicated by the blue bar. But that stiffness increase can be also cancelled or inhibited by the FGF19 or also obeticholic acid. So this is really suggesting or highlighting that potential for the clinical trials like condition in the dish. So by multiplying the genetic factors as well as the lifestyle factors or screening factors, we are able to conduct the human organoid-based trials or hot trials, which we named, for the efficacy screening in vitro in the future. And we are also developing a higher throughput form for measuring the stiffness measurements, which is not really showing up here. And also we are working on the sort of digital solution to be integrated to monitor the organoid in a wireless manner and so forth. But anyway, we are expecting in the future organoid trials can be very meaningful for the clinical trials. And as a last part of my presentation is focusing on the phase 3 application, which is namely transplantations. So I think organoid or organ butt transplantation is sort of intermediate approaches between the currently prevailing organ transplant as well as the hepatocyte or cellular transplantation. So this is the organoid transplantations. What we want to prove is to make a functional organ by transplanting very immature tissues or very immature organoid to see if we can eventually rescue the metabolic liver disease. So I think there are a couple of leading initiatives working on the transplantations. I think Tokyo Medical Dental University, which is also my second affiliation, but not my group, but different group is also working on the transplantation of organoid, initially targeting on the inflammatory bowel disease using adult stem cell organoid, and also the Netherlands or European group or our group or Cincinnati student is also working on the organ transplantation application for treating the currently intractable disease. So I think the next slide is just a simple view of what we want to do in the future. This is quite simple. So organoid manufacturing and transplant into the patients, hopefully transplant within a couple of years. So this is simple, but I think it's really challenging because number of different components for organ production has to be supported by the industry partners, because I think we need to have a clinical grade media, or we have to have a clinical grade culture plates, or we have to develop a clinical predicting preclinical animal model, of the small rodent model, or sometimes requiring the large animal model as well. And we have to develop clinical validation protocols by microscopic analysis as well as other biochemical assays. And probably I somehow need a robotic support for scaling up the process. So this is really, I think, very challenging and too much for the academic scientists. So we need a collaborative networks by working with the number of different industry partners to do that. So I'm just showing a couple of different examples of the collaboration with the companies. So this is our mass production platform by collaborating with the chemical companies called Takeda. So we have developed a clinical grade a micro well plate, which is an omni well, one well array plate platform, wherein we have the 20,000 micro wells are placed over the well plates. And we can make a very homogeneous, beautiful miniature organoid structures in a single culture time. So, and to the right, this is the collected human liver organoid from the pluripotent stem cell, just showing you the homogeneous tissue can be formed in a very large scale manner. So we have the mass production platform. And also we are working on the robotic companies to make GMP manufacturing facilities. So this effort was primarily led by Dr. Taniguchi, working in Yokohama City University, in collaboration with my laboratory. And as you see, I think this is also the movie, but we have the robotic automated machines to make our hundreds of organoid can be manufactured in a continuous manner, in an isolated manner, which is a clinical grade. So having that collaborations, we are hoping to transplant back to the patients within the next few years by initially targeting the urea cycle disorders. So for this particular applications, we are using the human iPS cells provided by the Kyoto University. I think as you are already aware, but Dr. Jun Takahashi at Kyoto University initiated the clinical trials using this particular iPS line for the Parkinson disease last week, actually. This is very quite exciting. But using the same iPS line they are using, we are also preparing for the clinical trials for the liver disease by developing a GMP-grade manufacturing process and transplant back to the patients to see if we can improve the clinical outcomes. So within the absolute indications, I think we have tremendous demand. So as I highlighted in background slides, we have over 4,000 of patients are just dying during the waiting period for the liver transplantations. But this is really underestimated, I think. And also we have multiple different countries waiting for the liver transplantations. So I think to be fair, I think currently overall over almost 100,000 of patients are waiting to die for the liver transplantations. So I think this patient population can be absolutely indication for even for the challenging organoid-based approaches. But within this sort of absolute indication, I think we can open up a very large, promising market for the future commercialization. So this is just a summary of my talk. I think in the future, I think we can transplant back to the patient, especially in a very end stage or a very chronic or organ failure condition, which we can certainly transplant organoid into the patients. But I think organ transplantation or cell therapy products is really expensive. So I think we need to narrow down the indications to really specifically targeting the patients who cannot really treat it by other modalities. But I think majority of the patients is benefited by the precision medicine application or drug treatment. So I think organoid is really meaningful to still for the drug screening or precision type of applications. So this is just a sort of overall big picture of what we are predicting in the future by using organ medicine applications. With that, I'd like to thank all my members in the laboratories as well as the collaborators, and thank you so much for your attention and happy to take some questions.
Thank you, Dr. Takebe, for that really amazing presentation. So we'll now begin the live question and answer portion of this webinar. So I'd like to remind you again, if you do have a question that you'd like to ask, Please do so now by clicking on the Ask a Question drop-down box located on the far left of your presentation window. Type your question in the box that appears on your screen and then just click the Send button. So with that, let's get started. So Dr. Takebe, we do have a few questions here. One of the questions that has come in is, what do you think are the technologies that will be driving your Organoid 4.0?
Hello. Okay, so you were asking about this question number 2?
Question number 8.
Number 8. Okay, technologies will be driving Organoid 4.0. So I think it's there are a couple of differences depending on the application. So for instance, I think drug screening application, I think technologies we are lacking is the sort of higher throughput screening system. So for instance, I think in our organoid model, we are sort of using the live imaging approaches or live stiffness measuring approaches to predict the steatohepatitis conditions. But I think still, the throughput is limited to test. I think at best, I think we can test the hundreds of compounds in the organoid level. But in a real world or in a real drug screening companies, I think they are testing over hundreds of thousands of compounds in a cell line model. So we need to increase the throughput extensively. So for the particular drug screening application, I think what we are lacking or what we need is for the higher throughput instruments to measure the organoid readouts in a higher speed. So with that, I think we are collaborating with the sort of micro robotics scientists or microfluidics scientists to make this happen. So I think depending on applications, but particularly for the drug screening, I think the microfluidic type of application is really important. But for the transplant application is really different. So transplantation is really critical, really driven by I think the mass production in a clinical grade. So just making tissues in a larger scale is quite easy, I think. But in a clinical grade manufacturing process is, I think, really challenging, because we need validation protocols, and also we have to manufacture in a very clean or very clinical grade and facilities to make sure they are pretty safe. And also we need to test all of the mass-produced tissues in an animal model to confirm the safety, to make sure we do not really develop tumors or uncontrollable or cancer-like tissues in vivo. So clinical grade manufacturing or mass production is really critical technologies we are still dealing with for the transplant application. So it's really depending on the application, but I think there are a couple of key hurdles still existing.
And how far away do you think that kind of technology is? Is this something you think we'll see in the next couple of years, another 5 to 10 years?
So yes, we are actually planning to transplant within 3 years from now. So we are almost in the final process of the manufacturing process as well as the validation process. So currently we are planning on the safety trials with the efficacy with the aim of obtaining the efficacy signals, preclinical efficacy analysis. We are planning on 2020 or 2021 to be initiated. So it's not really far future. And for the precision medicine, I think as you already know, the cystic fibrosis application is already initiated in Hans Klever's group in the Netherlands. So they're using the organoid tissues for model or predict the human condition in a cystic fibrosis derived organoid. And they are actually selecting the effective drugs and now treated by the drugs selected by the organoid. So for the precision medicine, I think it's already happening. So it's not really far future. I think it's a really tangible feature.
Okay. Thanks for that. And it's actually kind of a good segue to one of the other questions that is here as it relates to adult tissue-derived organoid models. As you know, some of the other models that are out there, there's an adult tissue-derived liver organoid model as well. Perhaps you could compare your model with that model and what you think the sort of the advantages and disadvantages are of the existing models today.
Thank you so much. I think this is really important. So I think we have good and bad. So for the iPS or ESL derived organoid model has an advantage for the multicellular configuration. So I think mostly the adult tissues derived organoid is epithelial cell structures. So we have a couple of cell types, but mostly are epithelial cell types. But iPS or ESL-based approach, we are able to co-develop supportive lineages, including the mesenchyme components and also immune components. And in addition to that, we have epithelial components. So multicellular configuration is, I think, important. But the downside of organoid from the iPS or PSC is I think, the maturation stage. So, the adult stem cell is adult derived, but iPS or ESL is from the embryos or egg stage. So it's really fetal embryonic stage. So functionality of the iPS or PSC model is a little bit immature or completely less mature than adult stem cell organoid. So for the drug screening application or some of the organ functions, is a little difficult to predict in a PSC-based model, but can do with the adult stem cell organoid model. But interestingly, I think for the transplantation, I think the adult tissue derived is a little difficult to engraft in vivo, probably because of the lack of supporting components, and probably because the immaturity is really important for the eventual engraftment. So I think, for the transplant application, we are intentionally using the immature or less mature organoid tissues for the transplantation to improve the in vivo outcomes. So there are many still arguable points for the PSC-derived or adult stem cell-derived organoid model, but each has a different advantage for the different applications.
I think we have time for just another question here. So a question that just came in is asking, regarding transplantation into liver disease patients, have you estimated just how many organoids you would actually need to transplant to have a therapeutic benefit?
Thank you so much. So currently we are planning to transplant over, I think, 100,000 of organoid per patient with the congenital disease for the kids. So which is equivalent to the 10 to the 8th or less than 10 to the 9th cell or number equivalent, hepatocyte equivalent. So this number is actually determined by the previous clinical trials of the primary hepatocyte transplantation actually implemented in the clinics. So for the hepatocyte transplantation, I think in the clinics, the physicians are transplanting over 10 to the 8th hepatocytes into the patient and already see the benefit from the disease conditions. So we are probably initially planning to transplant the equivalent number of hepatocyte transplantation, which is about hundreds of thousands of organoids, to initiate clinical trials. Thank you so much for asking the questions.
That is a lot of organoids.
Yes, it's a lot.
So with that, we'll bring this webinar to a close. Once again, I'd like to thank Dr. Takebe for his really excellent presentation here. And before we go, please note that today's webinar will be available for on-demand viewing through February 2019. And thank you all for joining us, and we hope you enjoy the rest of today's event.
Thank you so much. Bye-bye.